next speaker on the agenda is still on the road, unless I see him here. No, I don't think so. So therefore, we move on with the next. You see it here, right? Konstantina is the next speaker here, and you're here. I know, hiding. And let me just introduce you with a little bit more really good kind of background here, right? So today you are, as it says here, at NLOREM, and you are the Senior Director of ASO Discovery and Research at the foundation. And of course, as it shows on your CV, you have been working on RNA research for some time, right? Before that, you led the RNA and technology team at Amina Therapeutics. That was also about double-stranded RNA and so forth. Before that, you also did work on rare diseases, so it's not a new area for you. But also before you went to these positions, you had a significant career in academia. And when I was the people that prepared this for me, when I was reading through this, it's quite something here, right? So we're talking about the PhD came from the University of Oxford, but then there were some prestigious awards. One is called the Sir Henry Wellcome Reward for Postdoctoral Fellowship. And this was done in Edinburgh and in my home country in Berlin, in Germany, in Berlin at Max Delbrück Center. So quite amazing. But if this would not be enough, you also got a major award, which I think is called the Women's Science Award, which was, I think, probably sponsored by L'Oreal and UNESCO for this. So, and I'm sure there's more to find about you. So thank you very much joining us for this meeting here. Really welcome this. We heard earlier today that there were a few foundations that mentioned, and NLOREM, we are known anyway, the company, right? So, but we had this in Piotr's talk, and I heard it from Michelle earlier. So there's already collaborations going on with people that are here at the meeting too. Today we hear about what you guys do when, again, about personalized ASOs in, you call it nano-rare patients, right? So we have rare, nano-rare, uncommon, and I still pledge that at some point we all call this commonality somewhere, and we're talking the same thing. Welcome, and the floor is yours.
Thank you very much. An amazing introduction. Thank you. I'm extremely happy to be here, and yeah, what a wonderful event. And for the videos, would I just pause? Okay, that's wonderful. All right. Great. Yeah. So feel free to call me Nadina, everybody. I have an extremely long name. I'm Greek originally. So I'm leading all the research activities at the NLOREM Foundation. So essentially from ASO design, discovery, and also all our core mechanistic research approaches to really reach more patients with more efficacious ASOs and in a safer manner. First of all, I would just like to take a few moments to introduce NLOREM and just tell you all what we're doing and what is really our mission. NLOREM's mission is to really apply the efficiency, versatility, and also specificity of the antisense technology to charitably provide ASO medicines to treat patients with nano-rare diseases, and that means less than 30 patients worldwide for free for life. And essentially the reason that we can do that, and I'm very glad that I'm hearing ASOs here since this morning, is that really we leverage knowledge and expertise that has been around for about 35 years. And you know, our founder and CEO is Stan Kruk, who is the former CEO of Ionis. So we have really leveraged all this expertise from Stan Kruk, his colleagues, and everybody else on the antisense space. That has been amazing. And at the same time, while being in the shoulders of these giants, we really want to build continuous research innovation. This is where breakthrough will happen, right? And our ultimate goal is one and only one. We really want to find the best ASO possible for each patient. And this is what my team and I are really working on. First, I would just like to take you through what is our NLRM pathway to treatment. So the first part is essentially, I believe this is the pointer. Maybe. The first part is the research, and then we go into preclinical development, the regulatory stages, and also the clinical development. So essentially, my team is initially involved in also what we call the feasibility review, where we have the application evaluation and feasibility. So the physician sends us an application on behalf of the patient, and what we essentially evaluate, and this is, we also do it initially internally, but also we have a committee of really experts that really provide either ethics or clinical development, antisense technology, RNA, all of that, to really evaluate whether the ASO technology is the optimal therapeutic strategy for this patient. And after we have this committee and have these discussions, then we really take the decision if that application will be accepted or declined. If the application then is accepted and really we think that NASO is a suitable therapeutic approach for this patient, then this is where really we get to work. And this is where the ASO discovery and optimization takes place. So the very first thing that we do is we take the whole genome sequencing. This is one of the most important things for us, especially for allele selective programs, which I will go on into a minute. And also we obtain the patient cells. So everything that we do, and because the FDA, as I will say in a moment, we have this unique guidance for ultra rare or nano rare indications. We don't have specific steps, but we essentially have a very extensive in vitro discovery step, which we really use the patient cells for. And then we go into ASO design with our in-house AI-driven algorithm. We design the ASOs, we synthesize them, and then we really start the discovery. After that, we enter for about 20 top leads in vivo tox studies. And this is essentially the rat tolerability studies where we screen for tox. And then after that, this is essentially where really the preclinical development starts, where we have GLP toxicology, and this is where the GMP manufacturing and sterile fill and finish start. After that, and this is, we hold the IND, so we draft this massive document, which is called IND, and we essentially submit it to the FDA. And as you can see, actually, I didn't mention that in every one of these steps, you have around all the numbers of how many programs we have currently in each of these stages. And the very last thing is, of course, the clinical study and treatment. And at the moment, we have more than 30 patients that we are currently treating. And we have 16 patients that are actively on treatment, and we have more than 20 INDs submitted. And the important thing is that we have this in all 4 divisions of the FDA. So just all in all, and we started really, really small. So I'm really proud to stand in here and say that we have about 330 applications submitted today. We have more than 160 patient-directed drug discovery programs. Today, 25 INDs submitted, and the 4 divisions of the FDA are supportive right now. So just to give you a little bit of numbers and where we stand at the moment and how we're treating patients right this day, this is the NLRM regulatory clinical pipeline. So as I said, the number of INDs that we have submitted and all that is more than 20. The number of patients covered by the INDs is more than 30, and the reason for that is because some INDs cover more than 1 patient. The number of patients treated is more than 20 right now, and we're going to duplicate soon because actually my team is doing a great work in discovering a lot of ASO, so we have a lot of programs. The number of ASO doses that we have administered are more than 90, and the maximum duration of treatment is more than 2 years. And the most important thing of all is, of course, that we have pristine safety and tolerability profile. And this, of course, is one of them, is the most important thing. So really the current status of the clinical programs is that we have a very pristine safety and tolerability profile. 8 out of 9 evaluable patients show evidence of clinically important benefit. And I will show you in videos in a moment what that means. Sometimes a video is more than 1,000 words, which means that we are already seeing really important improvements in multiple domains in CNS, continued benefit with continued treatment, and very importantly, this is something that it was quite surprising to us because we start very low and very modest, and then we dose escalate. We really see evidence of CNS benefit at very low doses. We also see, in terms of kidney function, because we have some kidney targets as well, target reduction and stabilization of renal function, recovery of functions lost, demonstrating really plasticity of the CNS. So for some indications that we actually thought, oh, this person might be too old to actually treat, we find out that that is really not the case. That we think that the CNS is much more plastic than what we initially thought. And in terms of ALS, because we have quite a lot of ALS patients, we're really seeing halting of ALS disease progression. The important thing is, as I said, we have programs in all the 4 routes of administration: CNS, liver, kidney, and eye, and we're seeing important benefits in all of them. Now, what brings us to pristine safety profiles? It's really a big balance between what happens in research, what happens in discovery, and what happens in clinical development. So you really want a rigorous discovery, and this is exactly what we do. We screen thousands and thousands of ASOs to really find the one that is really the most optimal and best. So we really want to be very rigorous in that and really screen a lot in a very high throughput, high quality manner. But at the same time, on the other end, we want to be very conservative in our dosing and monitoring in terms of clinical. So what allows us to do that is a very deep and broad relevant experience in both ASO technology, drug discovery and development, clinical development and management of patients in clinical trials, and also our regulatory processes. We have an excellent relationship with the FDA. Our systems and processes are actually can be really scalable almost indefinitely. So we have really industrialized system and processes. We have multiple external committees for the preclinical data package review and clinical safety. As I was saying, this even starts from when an application comes to NLORM. It's not that we're just deciding internally. We have a lot of external people where we do a peer review. We really have a very strong commitment to caution, and also our research physicians are really outstanding. Some of them here at Radies and all over the US. We have a very close collaboration with them. And now I would just like to show you, this is one of our patients. This is a KIF1A. This is an allele selective ASO, which I will talk about in a moment. And I would just like to, this girl had a really severe disease with multiple seizures, neuropathic pain, couldn't walk, nonverbal. So I would just like to show you.
I'm ready, Sis. Oh, wow. That was...
Yeah, and then what?
And then when
Oh
my goodness.
See?
Look at that.
get up, you know when
Oh!
you
That was amazing.
I like that step though, but again. Boom, yeah. Ooh.
Dad, come on, I'll teach you how
Okay.
to stand
You
up.
teach me how to stand.
Throw an alley-oop. That's an alley-oop. Easy peasy, lemon squeezy. Oh.
Yeah, it's pretty incredible. So this is just boring words to just summarize what you just saw. This girl was, she was in a very bad state. This is a KIF1A disorder. As I said, very frequent and very severe seizures. We're seeing a significant reduction in seizure frequency and severity, significant reduction in neuropathic pain. She just wouldn't sleep because she would just cry because of pain the whole day. She's regaining, as you saw, the ability to stand up and work social distances independently. She couldn't do that before. She has a significant reduction in tremors, significantly improvement in communication with family. This is something that has been communicated to us by from her caregivers. and general significant improvement in the quality of her life. She even attended a sleepaway camp for the first time in her life. And of course, we have actually just published that in 2 publications. The second one that I would just like to show you is an ion channel mutation. This is SN2A. This is a boy that has actually, the ASO that we discovered and developed was actually administered here in Rades. This is a boy that has never walked in his life and had very, very severe GI dysfunction as well. This is a very severe disease, extreme seizures. This is an ion channel, so you can imagine this is very, very severe. This is an allele selective ASO as well. And he's walking independently for the first time. So this again summarizes what I was just saying. Reduction in seizures, he regains the ability to walk independently, decreased ataxia and choreoathetosis, and improvement in autonomic dysfunction, normalization of bowel movements, which is amazing for him and also for his caregivers as well. So all in all, I just talked to you before about what is happening with the clinical development. I would just like to show you as a whole what we do at Enlorem. And this is really our process. This is our ASO discovery and development process. It's an individualized approach and infinitely scalable. We really use, as I was saying, the efficiency of ASO technology in parallel with the FDA guidance for individualized ASOs to really provide a rapid, effective, and affordable treatments to nanorare patients. Everything starts with the whole genome sequencing and the patient cells. This is really the most important thing. This is where the ASO discovery begins, concludes that we enter the GLP toxicology. In parallel, we have to accelerate timelines, manufacturing, and sterile fill and finish. In parallel, our regulatory people at Enlorem, they start drafting the INDs and submit. We have the IND submission, IRB approval, which comes from where the physician is, and then of course we have the patient treatment. For the last part of the talk, what I would like to do is to take you through what my team and I are doing, and how do we end up with that optimal ASO? What does this look like? So this is essentially our process. But before that, I would just like to take a step back. And as I was saying, I'm extremely happy today that I heard ASOs multiple times. And I think the reason for that, the reason I think that this modality is really an ideal one for nanorare patients, and also importantly, why we do what we do at Enlorem and why we do it so well, is essentially because of this particular modality. For example, if you were to take, the really big guys at drug discovery, which is essentially historically, right, the small molecules and the monoclonal antibodies, you would know that the rules of engagement are not as well understood. Whereas with ASOs, with RNA targeted, we know exactly what are the rules of engagement. We know that in this particular case, the receptor, if you like, is the RNA. So you know exactly what you're going to get. After you design an ASO, you know what it will do. It will target that mRNA, it will hybridize like a zipper, and then it will degrade or it will block something. So since then the rules of engagement are very well understood and easy to use, it really means that we can also learn from previous drugs. Not everything is just like, sometimes with small molecules, we say change the method, change the drug. In this particular case, we have a broad transferability. The cost of drug discovery here is modest compared to other routes of drug discovery. The rate can be months. It is very, very selective. As I said, this is an RNA. This is just based on base pairing and hybridization. Very versatile. A lot of different mechanisms here that we can do. We can alter the RNA in many different ways. And also the important thing, and this is what matters, right? When you want to do breakthroughs and when you want to do things faster and better and safer, is really to advance the technology. And with ASOs, there is an extensive rapid advancement. And this is what we strive to do. very briefly, and I don't want to bore you too much with all the science and stuff, but a lot of people talk about gapmer ASOs, right? This is the RNase H1 ASOs. This is literally what is the drug. This is literally the ASO. And a gapmer ASO, this is what is used when you want to reduce or knock down an mRNA target. And essentially, we have what we call the 5-10-5 modality. So this is where you have the 5 bases at the wings at the very beginning there with the 2 black, which is RNA bases that are essentially or modified with methoxyethyl. And then you have in the middle there that orange part, which is the DNA. This is essentially where all the magic happens, because this is exactly what provides the substrate for a beautiful enzyme, very, very selective, which is called RNase H1, where when this orange binds your mRNA target, this is where the cleavage happens. And this is how you reduce the target. Now, you can do that in what we call a non-allele selective way. So you can reduce both your wild type, the good copy, but also the bad copy, the mutant, and that is what is not an allele selective. But the beauty of this enzyme also allows you to do it in an allele selective way, which basically means that you can leave your wild type untouched, but you can only target the mutant. And I will show you in a minute how we do that. Now, this is what we use when we want to degrade, when we want to reduce the target. We also have the other class of ASOs, which is called the steric blocking ASOs. This is an 18-mer, and as you can see, there is no orange here, everything is black. And the reason for that is because you essentially have only RNA bases, that they're all modified, the 2' methoxyethyl. And here the backbone is phosphorothioate. And something that I forgot to mention before is actually, you might ask, why do you need all of these weird chemistries? Why do you need to chemically modify the backbone or the RNA bases or whatnot? The reason for that is because this allows you to have a longer duration of action of the drug, less immunogenicity, and also higher binding affinity with the RNA. This is the reasons why we chemically modify ASOs. So you have different classes. So this would actually, you would sit back. So for example, when we receive an application for a patient, we sit back, we see the disease, and we're thinking, what is the best strategy for that patient? Do we want to reduce something? Do we want to reduce both the wild type and the mutant? Do we only want to reduce the mutant? Or do we want to block something? Do we want to use a different isoform? Do we want to alter splicing? And the reason that we can do that is because really we have, the ASOs are very versatile. So as I said, we can do an RNA degradation. These are the first 2 in here, either with RNase H1, ASOs, or you can have the siRNAs with AGO2, albeit a little bit more of a promiscuous molecule, not as specific. But also you can have mRNA modulation. So the splicing ASOs that I just showed you before in here can be used to either alter splicing or affect mRNA stability or switch between different isoforms. So this brings me to the allele selective ASO approach. And this is both of the ASOs that I showed you in videos before, they were using these kind of ASOs, and we use them a lot in a lot of diseases for the CNS, because essentially you don't want to completely reduce the protein, but you really want to keep one good copy, but get rid of the bad one. So the way you do that, and this really leverages the specificity of the RNase H1 enzyme, is you really try to guide the RNase H1 to target only the pathogenic mRNA with leaving intact the wild type. How do you do that? We have these little signs in a gene that they're called SNPs. You might have heard of them, single nucleotide polymorphisms. And this is why the whole genome sequencing is so, so important to us, because we take the entire gene and we really see where these SNPs are. SNPs are basically positions where your wild type and your pathogenic differ. And this is what we need, because we need to find these little differences so that we guide the ASOs in these positions. So the ASO can specifically, the RNase H1 can specifically degrade the pathogenic with leaving the wild type untouched. Now, we've done that. So what I've shown you is we've decided on the strategy. What do we want to do? Do we want to reduce? Yes, no. Do we want to block something? So after we have done that, then we are ready to essentially design the ASOs. And in order to design, we have our own in-house ASO design algorithm, which essentially really is the one that makes our ASO discovery highly efficient. And AI is a a buzzword right now. It's amazing. The thing that is important with AI is what kind of data you bring in. And the kind of data that we bring in our design software is essentially, we're taking all of these millions of ASOs that both us and also ASOs at Ionis we've been dealing with, all relevant cell and animal models, hundreds of thousands of patients, continuous learnings from the whole genome sequencing, because it's essentially what we are striving to do, to design better and better allele selective ASOs, and designing ASOs to reach more patients. And also at the same time, of course, there's so much information out there that is amazing with the technology, and there are a lot of multi-omics data sets that we're essentially putting in our algorithm to really make it better and better. And I'll just finish the talk with just showing you, how does it really look like? You know, when you screen, how does it look like? What kind of graphs are we seeing? So our process is essentially the very first part, and this is where it takes some time, is the primary ASO screen, because we screen about 500 to 1,000 ASOs. And for an allele selective approach, as you're going to see here, is the blue is the wild type allele where you want it almost untouched, and the red is the mutant allele where you want it reduced. So you can already see that there are some patches there. Oops. There are some patches there that we have some really, really good ASOs. And we have very important and very strict minimum criteria. So things that do not pass our criteria, for example, of 80 or less than, more than 80% of target reduction, it won't pass to the next step. The next step is really the epitome, as I would call it, of drug discovery for ASOs, which is essentially the dose response evaluation, where we do a 5.4-fold dilution to really scrutinize the efficacy, potency, and also allele selectivity of the ASO. And here, what we take in this step is we take about a 50 to 75, so the best of the previous step, and we essentially scrutinize what is the IC50. This is what defines the potency. What is the IC50 of the mutant? What is the IC50 of the wild type? What is the reduction of the mutant? Do we have the wild type untouched? What is the selectivity that we see? And here you can see here, they are in the red box, some ASOs that really stand out in this particular case. And finally, as we move down through the discovery path, we also do an in vitro, of course, of target analysis. This is initially done in silico, but then, of course, we also do that in wet lab. And we do that as many as necessary to make sure that we eliminate any off targets. And finally, of course, we're going to do the in vivo talks later on, but you really want to check, you wouldn't put ASOs that you know are not safe into animals. So there is an assay that has been established for years now, which is called the BJAB assay, which is essentially, it's a very good indicator, is done in B cells, which is a good indicator of innate immune activation. So the ones that really activate immunogenicity are, of course, thrown away. So you have essentially surrogates for that in a qPCR. And finally, you can see these really fancy robots that we have in the lab, and this is what allow us to really handle about 90 patient programs at the moment, even though we don't have a very big team. So this is all liquid handlers for high throughput screening. So as I come to the end of my talk, these are the conclusions. So we have a very highly experienced and functioning team in place to discover and develop optimal ASOs. We have the capabilities to do this rapidly in a cost-effective and high throughput manner. Our clinical experience is outstanding at the moment. The ASOs given via multiple routes of administration are safe. Our clinical benefits are seen across all organs, and very importantly, at low doses and really early in treatment in CNS. And we're really treating these patients today, and we are extremely proud and happy about it. These are all the publications to date. We try to publish as much as we can to have all of this out there. And really, thank you.